Welcome to my channel SP Sir's Classes. My name is Sabzar. If this is your first visit to my channel, please hit the subscribe button and notification bell to get notified whenever I post a new video on this channel. Before I walk you through the topic of this video tutorial, let me tell you that I have already uploaded this video on my channel, but the quality of that video was not good, so I decided to remake the video and to upload it again. So let us begin with the topic, that is biotechnology. I'll be covering various aspects of this topic, that is biotechnology, under two chapters. The very first chapter will be principles and processes, and the second chapter will be, of course, applications of biotechnology. So let us begin with the definition of biotechnology. Biotechnology is a process of using living organisms, their cellular components like organelles, molecules such as enzymes, DNA, that is genes, and other molecules to obtain the products or to develop the processes which are beneficial for the welfare of mankind. I hope you understood the definition of biotechnology. Okay, let us get a little idea how biotechnology works for the welfare of mankind. We know that beta cells of Langerhans of our liver synthesizes insulin, which is used by the body to regulate the sugar metabolism. However, diabetic patients are unable to synthesize insulin. Therefore, they are advised to take insulin externally in the form of injections. Now the question arises, where does this insulin comes from? Actually, this insulin has been made available to humankind by the biotechnology itself. Biotechnology has made it possible to transcribe and translate the human insulin gene within the prokaryotic organism like E. coli or other bacterial cells. How? Let us understand the process briefly from this very diagram. This is a human pancreatic cell, the genetic material of which contains the gene of interest, that is human insulin gene, that is right here. That has been isolated from the cell. We have another cell, that is bacterial cell, from which we have extracted this plasmid, which will function as vector. The interest of gene, that is human insulin gene, and the vector, that is plasmid, are ligated with each other to form recombinant DNA, which is then transferred into the host cell, that is right here. Within the host cell, this gene of interest, that is insulin gene, will undergo translation and it will form protein insulin. That insulin will be later on extracted and purified and ultimately packed into the vials and made available to the patients in the markets. This is how insulin production takes place with the help of biotechnology. Let us now talk briefly about the history of biotechnology. Paul Berg, the American biochemist who received Nobel Prize in Chemistry in 1980, along with Walter Gilbert and Frederick Sanger, is known as father of biotechnology. The other two prominent personalities in the history of biotechnology are Stanley Cohen, that's right here, and Herbert Bayer. These are the two scientists who achieved the triumph of preparing very first recombinant DNA. Few geniuses were working on a gene which were conferring antibiotic resistance to the bacteria Salmonella typhimurium. What they did, they successfully isolated the gene, antibiotic resistant gene, that is right here, from the bacteria, and then ligated this antibiotic resistance gene with the plasmid of another bacteria and formed the recombinant DNA. And then they transferred this recombinant DNA into the host cell. They found that the host cell has been conferred antibiotic resistance to by the insertion of this recombinant DNA. So this became the basis for the recombinant DNA technology, and recombinant DNA technology is the main basis for modern biotechnology. So guys, this is it for today's class. If you enjoyed watching this video tutorial and found it helpful, then don't forget to hit the like button, share it with your friends. In the upcoming video tutorial, we will discuss tools and recombinant DNA technology. Till then, take care and Allah Hafiz.